Hi, I am Laura. We are going to be talking about storing bioimage metadata for the REMBI specimen module. In this RDM byte, you will learn about REMBI specimen, the metadata you should be collecting to describe it, and how to store it. The first thing you need to define for the specimen metadata is the experimental status. Here you say if this is a control or a test. Next is the location within the biosample. This could be a coordinate, or a particular well in a plate, or a part of an organ. This may be different depending on the imaging technique or area of research. Then you should describe how the sample was prepared. Include enough information so that someone with experience in the field would be able to reproduce a sample by following the information you provide. So assume that they know typical techniques that are used and name them using terms from an ontology if possible. Only include lots of detail if you are describing a novel technique. Next, state how the signal that you are measuring with your imaging technique is being generated, the content and biological entities of different channels. This part is probably best described using an example, so we will cover this in more detail on the next slide. Here is an example of an experiment using a TIRF microscope. It is a control sample of cells whose plasma membrane is imaged within 100 nanometers of the cover slip. This is typical of TIRF. Next, the preparation method is described. Note that this method assumes that the reader will recognize the techniques used, the abbreviations, and the equipment described. It is not a long description. In this example, the signal in the sample is generated by fluorescent proteins, but this could also be a stain that has been added to the sample. Next is the channel content, which specifies the proteins used and the channels assigned in the image. Finally, the biological entities that have been tagged with the respective proteins are named. If you are using a stain, the equivalent would be to state what stain you are using in the content section and what part of the sample you are staining in the biological entity section of the channel description. Where you can, try to use an ontology for naming things. Now, about storing the specimen metadata. If you are using a template from a repository, continue to use it. Otherwise, I would suggest adding this data to the same file as you are storing the REMBI biosample metadata. It is not helpful to have lots of small files floating around, so if it's appropriate for your data, try to keep things in the same file. The main reasons for splitting files would be if it is getting too long, or if the metadata needs to be stored in different directories or locations in order to be kept alongside the relevant data. Please see links to additional resources alongside this video, as well as other RDM bytes in this series.